So now we're going to get into neurons. Neurons are the excitable cells, meaning they're going to experience electrochemical changes, right? Basically a change in the charge between the inside and the outside of the membrane. We call this depolarization and repolarization. And this is what's going to allow the neurons to send what's called an action potential. This is the signal that they're sending, right? We have resting potential. This is where we basically have the same voltage across the membrane, and that inside the cell is more negative than the outside. And that more negative charge is a negative 70 millivolt charge. So this is when the cell's at rest. It's not sending any signal. But the action potential is when we change. We go from that normal negative 70, and we're going to actually completely convert over to a positive charge, right? A positive charge instead before coming back down to our negative 70. And it's this change, and it's a rapid fire change that happens down the line that is going to allow us to send a signal. But before we get into the electrical activity, because we have a whole video on that just by itself, let's go ahead and get into the neurons, the basics, right? So let's talk about some of the basic things. We have the cell body. This is also called the soma, and this is where the nucleus, the organelles, like all that kind of stuff are found, right? Then we have extensions coming off of that cell body. We have dendrites. These are going to be shorter little projections that receive information. They're going to pull information in. And then the axons, which tends to be a single projection coming off a bit longer, that's going to send information out. So we have a one-way road. Information comes in through the dendrites, is processed in the cell body, and then exits and travels down the axon to go communicate with another neuron that comes next. We see there's a couple pieces. We see that the axon is going to connect to the cell body at what's called the axon hillock. We travel down and we get to what the very end, which is called the axon terminal. The axon terminal, we see all these little tiny extensions coming off. And at the end of these extensions, we have these little bulbs. These are the synaptic end bulbs, and these are going to be where we have a bunch of neurotransmitters. We'll talk more about those at the end of this chapter. We have a whole video on it specifically. We generally have a myelin sheath going down. Again, remember, Schwann cells will make that in the peripheral side. Oligodendrocytes are responsible for that in the central side. But this is our basic generic neuron, right? So let's talk about the axons. These axons can be really short, or they can be really long. And when we say really short and really long, we mean we're talking about from short being a couple millimeters to an entire meter. That's about 3 feet, right, in length. These axons are really long. There's some that will travel from your spinal cord, right, area all the way to an organ. Literally from the spine all the way to the organ itself. So if we're talking about the adrenal medulla that is directly innervated, that is a single neuron that is reaching out from the spine to the adrenal gland. That's wild when you think about it. And so they vary in length, and we have substances that are transported in vesicles along the axon through microtubules. So there's actually little highways. There's like microtubule highways to help move things from the cell body where all the organelles are down the axon to the axon terminal. So it seems a little strange because you're like, how the heck does the cell maintain itself if it's 3 feet long? Even if it is really skinny, we have little microtubule highways running through there that little motor proteins can carry things up and down along those tracks. When we go from the cell body to the axon end, this is called anterograde, right? Because we're going forward, whereas axon to cell body, we call this retrograde axonal transport because we're going backwards, right? Like retro. So now let's go ahead and talk about that myelination. We talked about the oligodendrocytes, we talked about the Schwann cells, but we didn't actually talk about how do they myelinate? What is myelin sheath? Why have it, right? What's the purpose of this in the first place. So let's talk about the myelin sheath consistency first. Like, what is it made of? So this is going to be made of myelin, right? Myelin is a very lipid-based substance, a fatty substance that is basically 75% lipids, 25% proteins, right? I'm not worried about you memorizing that, but you should know this is a lipid-based component. This is to electrically insulate the axon. So think of it like how if you have wires, you don't just want to have the raw wire, you have some sort of cabling, you have some sort of protection over the wire itself. The myelin sheath kind of acts like that in a way. And this again is going to insulate, and because of that we can increase the speed of the impulses as well. Considering we have to sometimes travel up to 3 feet very quickly, that becomes really important, right? But we notice it's not completely covering the axon. There are little gaps, right, in between. We call these the nodes of Ranvier. And the nodes of Ranvier, right, these little gaps, this is going to help with propagating what we call action potentials, or APs. So action potentials via what's called saltatory conduction, right? Saltatory conduction. This saltatory conduction is actually going to allow us to speed up this process. So we can basically, the signal that's being sent, the action potential, can jump from kind of like the covering to covering, kind of jump that gap, right? And this is going to help speed us up. So you can imagine if the myelin sheath is providing us with this insulation and this way of making the signals travel faster, that losing that myelin sheath can be very problematic. So here's a couple conditions that are caused, right, by the destruction of the myelin sheath. And we're usually looking at autoimmune, right, with both of these. So Guillain-Barré syndrome, this is where the T cells are attacking the Schwann cells. This is a peripheral nervous system problem. So we get muscle weakness, we get tingling, so using your limbs would become much more difficult. Whereas multiple sclerosis, or MS, this is where the oligodendrocytes are targeted, so the myelin sheath in the central nervous system is impacted. And this can really cause a wide variety of problems depending on which parts of the central nervous system, like which parts of the brain are affected at that moment. It can lead to problems with vision, it can cause pain, it can cause fatigue, it can cause muscle weakness, problems with coordination and balance, tingling, problems with speech. I mean, you name it, it can be affected by MS. Now, because this is such a dense chapter, I'm not going to be so worried about you remembering all these different diseases. So when we're talking about the diseases, just keep in mind we're focusing on just kind of explaining why these different functions, why these different parts are important, and that just giving you an idea of how the loss of those functions, the loss of those components can affect a person. But for the sake of an exam, I'm only going to really worry about your understanding of the normal function of things. Now let's move on to the types of neurons we can find. So we're going to talk about some different structural classes. Right? So these are basically how many pieces are sticking out is kind of the way we structurally class them. So here we have a multipolar neuron. So we have lots of dendrites. They're all over the place, right? Lots of dendrites and then one axon. So multipolar because we have more than two, right? They're coming off, we have extensions coming off all over the place. With the bipolar neuron, we have one dendrite to the cell body, right? And then down the axon, to the axon terminal. But we see that the cell body is sandwiched in the middle, right? And there's two extensions coming off. With the unipolar, it's similar to the bipolar. We have the dendrites, and then we come down the axon, but the cell body is actually kind of off to the side. So this length is kind of one continuous piece. We don't have the cell body kind of interrupting it the way we do with the bipolar neuron. So you should be able to kind of explain these in your own words, or be able to pick them out or draw them yourself to kind of describe the structural classes. And then sometimes we have like just really special ones. These ones are just kind of different and not as common as the other three we just saw. So like the Purkinje cells, these ones are ones we see inside of like the cerebellum. These ones have like this crazy network, very elaborate, right? Kind of design, right? Kind of structure. And so they're going to help with coordination, which makes sense that they're going to be connecting and talking to tons of different neurons, which makes sense when your job is to coordinate. And then the pyramidal cell, this is going to be in the cerebral cortex, your hippocampus, your amygdala, and whatnot. This is for visual processing and motor function. And we see we have almost a triangle-like appearance when we're looking at the flat two-dimensional picture. The dendrites kind of extending off every which way, and then the one axon coming down. So we can also classify them based on the direction of the nerve impulse. So these neurons can also be classified based on the direction. So we have like motor neurons that are going to carry signals just from that, in through the dendrites, through the cell body and down the axon. Interneurons are going to process that information, and sensory neurons are bringing it in. So motor neurons are going to be efferent, interneurons process in the middle in the central nervous system, and sensory neurons are afferent. And so we just see this directional, kind of chart here. And now if you notice, looking at that unipolar sensory neuron up at the top, notice the dendrites have been modified in this case. So the dendrites in these sensory neurons can be modified based on what they're trying to sense. So for example, think back to the skin. When we were talking about the skin, you saw like Merkel cells and like all these different like corpuscles and whatnot. And all of those had a shape to them, and those were the modified dendrites. Right? That then connects us to the interneuron, which is going to be in the central nervous system, processing that information, which then sends that signal to the motor neuron out for the afferent direction through to kind of cause an effect. Can also base them on their function. So motor neurons are going to be classified either as somatic or autonomic. So somatic, these are going to be involved with your skeletal muscles, both voluntary control and your reflexes. Whereas autonomic, we're thinking of all the other things. So like your smooth muscle and the blood vessels, your cardiac muscle, your glands, right? All those types of things that you don't necessarily have to think about in order to enact some sort of action in them. And with the autonomic side, we have two sides. We have the sympathetic and parasympathetic. Sympathetic being fight or flight, and parasympathetic being rest and digest. We'll talk more about the autonomic split in a totally different video. When it comes to sensory, same thing, we have somatic and autonomic sensory neurons. Somatic are going to be things that you think of like your general senses, so like touch, pain, pressure, temperature, proprioception, which is like your orientation or like your balance, and then special senses like seeing and hearing. And autonomic is going to be things like the sensors coming from your organs, from your blood vessels, from your muscles, and certain special senses on top of that. And when we look at collections of nervous tissue, we have very specific terms. Now, we have ganglia and nuclei. Both of these are going to be the collections of the cell bodies. So like these little swellings, right, where we have these cell body collections. But the difference is where we find each. Ganglia are found in the peripheral nervous system. Nuclei are found in the central nervous system. They're generally the same thing for the most part, but that's the difference. Nerves and tracts, we see the same thing. Nerves and tracts are both bundles of axons. Nerves, though, are peripheral. Tracts are central. And then we do see two types of matter in the central nervous system, white matter and gray matter, aptly named for the colors they actually appear. The white matter is white because it has all those myelinated axons. Remember that myelin is a fatty lipid base, and so that's where we're getting that coloring from, whereas the gray matter, we're looking at the dendrites, the cell bodies, and unmyelinated axons, which gives it that grayish color. So if we look at how it's distributed, again, here they're using a beige color, but we see the darker colors are going to represent the gray matter, and the lighter beige is going to represent the white matter. We notice in the spinal cord, the gray matter is more central, more in the middle, and the white matter surrounds it. But when we look at the brain, we see it's reverse. The gray matter forms the outer edge, and the white matter is deeper in. Now, when it comes to neurons, neurons are a little different from a lot of other tissues we have. They cannot necessarily easily regenerate. Some can, most, especially when we're looking central nervous system, cannot. And so instead of relying on regeneration, we rely on plasticity. Now you may be asking, well, what's the difference? Regeneration means you're replicating or repairing. A neuron is damaged, so we can either fix it or we get rid of that neuron and we replace it by dividing a neuron nearby. Plasticity, this is where rather than replicate or repair the cells, we're just going to create a new neural pathway that goes around the damage. All right, that's the difference between those two. So if there's an injury or disease that damages neurons, if we can, we'll try to regenerate, but in a lot of cases, regeneration is not possible. And so plasticity is what we rely on instead. Now, if we're looking at the peripheral nervous system, we can repair some neurons. It just depends on what part's damaged and how. And so if the cell body's intact, we may be able to actually fix it. So the first thing we go is chromatolysis, where we're basically redistributing what are called Nissl bodies, which is just the rough ER, which is going to help produce proteins. And this is because we're going to need a whole lot of new proteins. So we're redistributing them so we can make more proteins. Then Wallerian degeneration. This is where we remove the axon fragments. Basically, we remove the portions of the damaged axon, and then we regenerate that axon using proliferating Schwann cells. Right? In the central nervous system with the brain and spinal cord, we don't have this happen, and so we rely on plasticity in order to correct any damage. And sometimes we can, sometimes we can't, right? It just depends.